Welcome everyone. Today we will discuss about life cycle of Plasmodium vivax. Plasmodium, it is a protozoan parasite which is responsible for causing the malaria in man. The life cycle of Plasmodium vivax is digenetic. It means that it requires two hosts for its completion. One host is vertebrate host, that is man, and another host is a invertebrate host, that is female anopheles mosquito. The Plasmodium vivax is reproduced by sexually as well as asexually. The sexual phase of life cycle, that is schizogony, takes place in the body of man, while sexual phase of life cycle, that is sporogony, takes place in the body of female anopheles mosquito, and therefore female anopheles mosquito is called as a primary or definitive host, while the man is called as a secondary or intermediate host. This female anopheles mosquito, it is act as a vector which transport the plasmodium from one person to the another person. This diagram showing the asexual cycle as well as sexual cycle of Plasmodium vivax. Asexual cycle is occur in a man in liver cell and the RBC, while sexual cycle takes place in the alimentary canal and salivary gland of female anopheles mosquito. A sexual phase of life cycle takes place in a man. So first we will discuss about the life cycle of Plasmodium vivax in a man. This asexual phase of life cycle of Plasmodium vivax is completed by the schizogony, which is differentiated into the liver schizogony and erythrocytic schizogony. Liver schizogony has the two phases, pre-erythrocytic phase and exoerythrocytic phase. This diagram showing the asexual cycle of Plasmodium vivax in man. When an infected female anopheles mosquito bites a healthy person to suck the blood, it releases some saliva into the bloodstream of that healthy person. Along with the saliva, thousands of sporozoids enters into the bloodstream of the man. This sporozoid, it is the infective stage of Plasmodium vivax. If you want to study the detailed structure of the sporozoid, you can watch my previous video, that is structure of sporozoid. This parasite circulate in the blood about 30 minutes and then disappear from the bloodstream and do not appear there again until the 6th day. During this period, they enter the liver cell where they start the asexual method called as schizogony. In the liver cell, sporozoid grow to a large and round schizon. The schizon is divided by the multiple fission and form 1000 to several thousands of small spindle-shaped cell called as merozoid. Then that schizon ruptured and releases the cryptomerozoid into the liver sinusoid. So this phase of asexual multiplication is called as pre-erythrocytic cycle. This cryptomerozoid has a compact nucleus and no pigment or vacuole. These cryptomerozoids are resistant to the host body and immune to the medicine. Now this cryptomerozoid has a two-way. One, it may enter into the bloodstream and infect the red blood cell to carry the erythrocytic cycle, or it may infect the fresh liver cell to carry the exoerythrocytic cycle. So now we will discuss about the exoerythrocytic cycle. This exoerythrocytic cycle is begin when the cryptomerozoid of pre-erythrocytic cycle infect the fresh liver cell. That cryptomerozoid then grow into the schizon. Schizon divide to form a several thousands of merozoid. Merozoid of second generation is called as metacryptomerozoid. There are two types of metacryptomerozoid, micrometacryptomerozoid and macrometacryptomerozoid. Micrometacryptomerozoid, as the name suggests, they are smaller in size and more in number, and they invade the RBC and continue the erythrocytic cycle, while the macrometacryptomerozoid are larger in size and less in number. They remain in a liver cell and attack the fresh liver cell and continue the exoerythrocytic cycle. Now we will discuss about the erythrocytic cycle, also called as erythrocytic schizogony. The erythrocytic schizogony, it is the third multiplication stage of the schizogony. It is begin when the RBC are attacked by the cryptomerozoid of pre-erythrocytic cycle or micrometacryptomerozoite of exoerythrocytic cycle. After entering into the RBC, the micrometacryptomerozoite becomes rounded and disc-like in a shape, which is now called as a tropozoite. This tropozoite grow in a size by ingesting the hemoglobin present in the RBC, due to which vacuole is appeared in the parasite. This vacuole gradually increase in a size and push the nucleus to the one side in the peripheral cytoplasm. This stage referred as a signet ring stage. As the parasite grow further, the vacuole disappear and the shape of the parasite becomes amoeboid. That stage is called as amoeboid stage. After the active feeding, the tropozoite grow in a size and becomes a round called as schizont. The larger schizont makes the erythrocyte to become a very large. Schizont contain the yellowish pigment called as hemozoin and the scrupulous granule. Then that schizont undergoes the multiple fission and form a 12 to 24 oval-shaped merozoite. This phase of asexual multiplication is erythrocytic schizogony. At the end, the erythrocyte are burst and releases the merozoite along with the hemozoin. That hemozoin is responsible for the occurrence of the malarial fever in the host. As the RBC bursts, the merozoite releases in the plasma. Some merozoites are destroyed by the WBC, while some merozoites infect the fresh RBC to start the erythrocytic cycle. Some of the merozoites go from blood into the liver cell and start the asexual multiplication in the liver cell, which is called as a post-erythrocytic schizogony. While some of the merozoites forms the megagametocytes and microgametocyte. For the completion of the one erythrocytic cycle required 48 hours in the Plasmodium vivax. As you can see that in the erythrocytic cycle, RBC breakdown, and this is the reason the malaria patient becomes anemic. So this is all about the asexual cycle of Plasmodium vivax which occur in a man. In the next lecture, we will discuss about the sexual cycle of Plasmodium vivax which occur in a female anopheles mosquito. Thank you for watching.